Hello students, this is Dr. Faizan Mirza. I'm discussing with you the effect of wavelengths on the rate of light penetration, how that can be followed in a chemical reaction or in an experiment. So what are we doing in this experiment? What we'll be using chloroplast isolated in a buffer solution. We'll wrap them with an aluminum foil and we'll allow light to pass by removing the aluminum foil and see how the color of DCPIP gets discharged, because the light dependent radiation uses light energy as a source of energy. So what we'll do, we'll take isolate of chloroplast taken from fresh leaves using centrifuge. We'll be taking these isolates and the sediment of the centrifuge tube will be collected, and these chloroplast sediment will be taken and these will be transferred into a test tube, and this test tube will then be placed together with isotonic buffer, and we'll add isotonic buffer to the chloroplast extract to make a mixture here. The test tube will be kept wrapped in aluminum foil to keep it away from light source. So this is how the centrifuge tube would be, and this is how the aluminum foil will be wrapped around it. Now what will we do? We'll take, for example, 10 ml suspension into a separate test tube and again wrap it in an aluminum foil, and I'll add 1 ml of DCPIP, which is blue in color. DCPIP, it is used as an indicator here. It gets reduced when light is shown to it. So I'll take DCPIP, which is blue in color. I'll keep the light source of single wavelength at, for example, 20 centimeter from the test tube. I'll expose the contents of this to the light source, and I'll note down the time taken for the DCPIP to decolorize or turn green. So this is the DCPIP, this is blue in color. I'll use, for example, red color source. I know the wavelength of red color. I'll mention the wavelength that I've used. I've placed it at a 20 centimeter distance. I'll see that the color of the color gets discharged. I will repeat this experiment 3 times with fresh contents of same quantity to calculate the mean, and then I can calculate the rate the rate of the rate of light dependent reaction by dividing 1 by time, and the time can be calculated how long it took for the color to decolorize. I can use a timer when I start when I initiate the light being shown onto this particular test tube. And this is how this experiment can then just go on. I can repeat this for all visible light wavelengths, and for each wavelength I can just plot a graph of how the wavelengths are taken. They can be taken on x-axis against the rate of photosynthesis, not the rate of photosynthesis, the rate of light dependent reaction. Yeah, it's worth mentioning this is not the rate of photosynthesis. This is not complete photosynthesis. Sugars are not being produced here. This is just the light dependent reaction, and we are using DCPIP here to see how the rate of light dependent reaction changes with the different light source. I am keeping different light sources here, and for every light source that I'll keep here, I have to maintain the distance of 20 centimeters, so distance stays the same. If I just need to change, for example, if I'm not, this is the effect of wavelength. If I want to change the experimental technique and I want to note down the effect of light intensity, what I can do, I'll use the light of one wavelength, for example, just red color, and I'll just start to place it at different distances, 10 centimeter, then 20, then 30, then 40, then 50, and so on and so forth. And I can see how the rate of light dependent radiation varies with the changing of distance. Again, the same protocol will be used, that which I have already described. That's it from my side. Thank you so much.